Okay, so maybe we can start our housekeeping notes. Hello, everybody. Thanks for joining us today. As usual, we start with some housekeeping notes, and then we have the presentation of our speaker, Dr. Feinberg. So today, we are very pleased to have Professor Feinberg from Carnegie Mellon University. He's talking about fresh 3D bioprinting, and I see that the title here is not that correct, but we have the title that Dr. Feinberg will share with us. So this talk will be recorded. Dr. Feinberg give us a permission to post this talk on YouTube channel, and then we have also Q&A box. As always, you can put your questions during the talk in the Q&A box, and also there is a poll section on the bottom of your screen, so you can take the poll to kind of give us feedback to improve the quality of the e-seminars. So by that. As always, also, and our next seminar is going to be Mr. Scott Phillips, from CEO and founder of Starfish Medical, and he will share with us the story of starting of the Starfish Biomedical companies, and next Wednesday at noon. And then if you have any questions or concerns, and you want to get any updated information about our upcoming seminars, follow us on Twitter, and also you can contact the team, Dr. Akbari, myself, and our coordinator Wahid. As always, we cannot run these seminars without the help of our sponsors. Our continuous sponsor, Montreal Transmedic Institute, which aims to support the accelerated development of next generation medical technologies applied to three broad categories of disease, for example, cardiovascular, neuromusculoskeletal, and cancer, in order to facilitate their implementation in the health system. And also they aim to train the next generation of medical technology professionals and make the innovation in life sciences and engineering a source of wealth for society. We also would like to thank the sponsorship from Cell Inc. company. Cell Inc. is creating the future of medicine by providing technologies, products, and services to create, understand, and master biology. While aiming to create the future of medicine, Cell Inc. has focused on three application areas that are essential to cell culturing: bioprinting, analysis, and liquid handling bioprocessing. With the combination of persistent passion and innovation, Cell Inc. accelerated the genesis of the bioprinting industry by bringing bioprinting technologies from the lab to the clinic with unprecedented speed. In doing so, Cell Inc. has transformed its technology into a globally recognized pioneer of the 3D cell cultivation system in pharmaceutical research and drug development. We thank Cell Inc. and Transmedic Institute. By that, I would like to introduce our speaker, Professor Feinberg from Carnegie Mellon University. Dr. Feinberg is a professor in the Department of Biomedical Engineering and Material Science and Engineering at Carnegie Mellon University. He is also the principal investigator of the Regenerative Biomaterials and Therapeutics group. His group develops materials-based engineering strategies to control the self-organization and assembly of various cell types into tissues. The goal is to understand the emergence of higher order function in biological systems, especially in ophthalmic, neuromuscular, and cardiovascular systems. Current research project in his lab includes biomechanics and mechanobiology of fibronectin laminin collagen nanofibers, development of new methods for fabricating ECM scaffolds, engineered basement membranes for regeneration of the cornea, endothelium, and cardiac tissue engineering using 3D structured ECM scaffolds, 3D bioprinting, and advanced biofabrication approaches. Dr. Feinberg earned his bachelor in materials science and engineering with an option in bioengineering from Cornell University, followed by his master and PhD in biomedical engineering from the University of Florida. He completed his postdoctoral training at Harvard University in the School of Engineering and Applied Sciences before he moved to Pittsburgh. Most recently, Dr. Feinberg was the recipient of many prestigious awards, including National Institutes of Health Directors New Innovator Awards in 2012, George Thalman Ladd Faculty Research Award, Carnegie Institute of Technology 2013, and National Science Foundation Career Award 2015, and also Arthur Hammerschlag Career Development Professorship 27. So Dr. Feinberg currently holds more than 20 patents and patent applications and is a member of the Materials Research Society, American Chemical Society, Society for Materials, Biophysical Society, Biomedical Engineering Society, and the American Heart Association, among others. He is also co-founder and CTO of Floyd 4 company, a startup focused on soft materials and 3D bioprinting. By that, I would like to invite Dr. Feinberg to share his screen, and the virtual floor is yours, Dr. Feinberg.
All right. Well, thank you, Dr. Akbari and Dr. Spoji, for the invitation to speak today. I really appreciate it. Thanks for that really kind introduction. You reminded me of a few things I'd even forgotten at this point, so that's always good to be reminded. Let me get the screen share up and running. Excellent.
Yeah, we see.
Perfect.
Yeah, perfect.
All right. So, well, I really appreciate the opportunity to speak with you all today and share some of the work we've been doing in my lab at Carnegie Mellon University in Pittsburgh. We've been here about 10 years, and really, we focus on a lot of things. As was mentioned, you know, kind of a key focus is really, how do we build tissue? How do we create environments that are instructive to cells to help guide the assembly into more complex structures, and ultimately get to the point where we can build tissues and then one day organs for a wide range of therapeutic applications. Fresh 3D bioprinting is one of the technologies that we've developed to do that, which I'll really go through in this talk and kind of describe how we got there, where we're going, and some of the new things that'll be hopefully coming out soon. And then, you know, as was mentioned, we've also got a startup. I'm also CTO and co-founder of Fluidform, which is commercializing the technology. So that is one of my conflicts, which I'll cover at the end. So obviously, please, you know, just want to be very transparent. So what you see on this very first slide, though, is really one of the culminations of this vision, which is to be able to 3D bioprint biologic materials and one day cells at the scale of full organs. And this is a paper that just recently came out in Science, in ACS Biomaterials Science and Engineering, where we've actually printed an allogeneic heart scaffold, no cells, with scaffold at full organ scale, which I'll talk about how we got there throughout this talk. But before we do that, I kind of want to take a step back and kind of talk a little bit more big picture about what we're talking about. So we think a lot about the ECM extracellular matrix. We think about cells, and we think about morphogenesis. when you want to talk about biomaterials, what are the biomaterials of the human body? Well, for really almost every tissue and organ, that's the extracellular matrix. This is, by mass, predominantly collagen, but it also includes a wide range of other proteins and molecules, things like laminins and fibronectin, which are other ECM molecules. You have proteoglycans, you have things like hyaluronic acid, you have growth factors that integrate and immobilize into the ECM. And cells are uniquely really built to both secrete and build the matrix and remodel that matrix. And so we really look at how can we use the extracellular matrix as a biomaterial in the same way that the body does. We feel like if we could do that, then we could really start to rebuild tissues in a way that hasn't been possible to date. We're also really inspired by embryonic development. And so this is just a phase image in the dish of a zebrafish embryo. And what you're looking at is basically that larger dark brown area shown here is the, and let me see if I can't turn on the laser pointer. There we go. Yeah, so over here, this is the yolk sac, and then this is the fertilized egg after the very first binary division. And what I want to show you is the zebrafish developing over the first 24 hours. So I'll play this video. What you're seeing now is those two cells are dividing into smaller and smaller cells through binary division. Because remember, the initial fertilized egg is actually a really large cell. So the first thing you do is you get these divisions. And then as that happens and the cells get smaller and smaller, they start to migrate around this yolk sac, almost like migrating around the outside of a balloon. Now, as they contract against the yolk sac, they generate mechanical tension, and that allows the cells to start to organize and pattern into basically the animal. Now, as the cells are dividing, they're also specifying and differentiating, but they're also secreting and assembling the extracellular matrix at the same time. And at this point, we can start to see some of the somites forming here that'll form some of the internal organs. You can see the eye is quite visible here. And then as soon as the skeletal muscle forms in the tail here, you'll actually see the animal start to twitch as that muscle tissue becomes active. And so this is all happening in 24 hours. This is the zebrafish takes about 3 days to undergo development. So we can think about this as roughly like the first trimester of perhaps human development. And I think what's really interesting here is that, again, this started as a single cell, and as the cells divide and secrete matrix, they build all of the structure. This is all genetically encoded, right? This entire process is based on the DNA that drives the instruction set for this entire process. And so what we want to do when we look at developmental biology is extract engineering design principles that we can then use outside of normal development, more in a synthetic context, but to build these tissues, again, inspired by embryonic development. But how do you do that? So this talk is focused on 3D bioprinting as one way to get us there. And this is actually a cartoon and illustration if you just Google 3D bioprinting. But it really shows the basic concept that you have basically your normal 3D printer, right? A 3-axis machine that has a cartridge for cells, for biomaterials. You have a CAD design, perhaps based on MRI imaging of a person's, say, unique heart structure, and that you can hopefully just print or press print, and the whole thing is printed layer by layer over time. So that's the vision, right? And the idea is that you can hopefully precisely place cells and biomaterials anywhere in 3D space, exactly the way you want that to recreate the structure. That's the vision. I think it's been a little bit overhyped. In fact, I think sometimes it's hard to tell what's reality versus science fiction. So I always like to show this quick image of the replicator from Star Trek. And if you remember, the replicator typically was what they used to create food from molecules that magically assembled. But even in Star Trek, you very rarely saw, if ever, actual living tissue being created in this process. And I think that speaks to, even in science fiction, I think what the challenges are of creating functional living tissue. I also like to show this hype curve, because I think it's important to understand 3D bioprinting is an emerging technology. It's incredibly exciting with a tremendous potential, Otherwise we wouldn't be working on it. But we're still very much in a hype period, right? If you haven't seen these hype curves before, and this one happens to be from 2018, there's an innovation trigger, there's a peak of overinflated expectations, there's this trough of disillusionment, and then a slope of enlightenment, and then a plateau of productivity. So a lot of new technologies go through this. And this is all of 3D printing. But if we look at bioprinting, 3D printing of medical devices is right here, still on this hype curve. The 3D bioprinting for life sciences, of human tissue, and then also of organs and transplants. And then these little icons actually indicate how far off they are. So, medical devices, 2 to 5 years, are actually already here now. So medical devices have started to transition. But things like bioprinting of living tissue is still in this 5 to 10 year timeline. And bioprinted organs are more than 10 years plus. And I think this is all accurate. I think it is our job as scientists in the space to be, enthusiastic champions of this technology, but also responsible in the sense of being realistic about what we can achieve, when we can achieve it, and really being rigorous with our scientific process. And so I just like to kind of frame where I think 3D bioprinting is, because it deserves all the excitement that has been kind of lauded on it, but it's also a technology that still needs many years of development to reach that promise. So I want to kind of get into exactly what we've been trying to do in the space of 3D bioprinting and why. And what we've really been trying to address is this problem of printability, specifically for hydrogels and cells. So if we look at a normal 3D printer shown here in the upper left of the slide, this is your normal plastic printer that many of you are familiar with. And the reason this works is that it obviously prints layer by layer by layer, but by extruding plastic and then letting it rapidly solidify, it becomes very rigid and solid. It's stable in 3D space. That makes it very straightforward to come down and put another layer on top because the layer below it is stable. Now that is not the case with many hydrogels and cells, because these are materials that have a very low elastic modulus. They typically are not able to support their own weight in air. And shown here is some hydrogel printing. As you try to print additional layers up, that is really, really difficult to do. You lose fidelity because when you put down a layer, it moves. And so it's challenging then to put the next layer on top. And as you build layers, that just increases your error. If you also have living cells, they have the potential to actually move on their own too. And so that's a whole nother layer of difficulty. So how do we print hydrogels and cells? I like to kind of talk about this challenge with the Salvador Dali painting, which shows these kind of deformed clocks. And the problem of trying to build a complex living biologic system out of soft materials that move is that you at least have to hold everything in place during the assembly process, even if that system is deformable and moves after the assembly process. So the analogy I use is, imagine you had a pocket watch that you disassembled. You had all the gears and springs laying in front of you, and your job was to put that pocket watch back together. I think most of us could start to figure out at least how some of those gear assemblies fit back together to rebuild that watch. Probably not the whole watch, but we could understand how pieces go back together. But now imagine you had to build that watch and all those gears and springs were soft and flexible and floppy, and they didn't hold their shape. How could you possibly rebuild a gear assembly with materials that don't hold their shape, right? And so again, we're dealing with materials that very much have the mechanical properties of something like Jello, which is easy to make into a block or a mold, but that's not what we're trying to do. We're trying to turn it into something complex, like say this CAD model here of liver vasculature. And I think we can all appreciate that if we tried to make something complex like the Jello mold into something like the liver vasculature, it would collapse under its own weight. So how do we do that? So my lab is by no means the only ones working in this space. There's obviously many labs doing amazing work really across the globe. I'm just going to highlight a few, obviously, in the interest of time. But there's a couple different strategies. So this is work from Tony Atala's lab at Wake Forest, where they're trying to print soft hydrogels. And the way they do that is they print a PCL frame, shown here, kind of like the lattice you would see, or a scaffolding like you would see on the outside of a building as it's being constructed. That PCL lattice is basically provides mechanical support, and the hydrogel, the cell-laden hydrogel, is basically extruded inside of the pores inside of this PCL lattice. And this is one way to create a rigid support material that can hold up a softer hydrogel. And they've used this and published it in Nature Biotechnology and other high impact journals. This is some work by Jennifer Lewis, who's really pioneered this approach of using a sacrificial pluronics filament, which is relatively straightforward to print. That's shown here, printing a large kind of a cube of this lattice structure. And then they can cast a hydrogel around that. And then using a temperature, a thermal reversibility of the pluronics, actually dissolve and wash out the pluronics, which leaves a vascular-like network inside of that hydrogel cast around it. And they've really done pioneering work creating larger vascularized structures with this approach. Published in Advanced Materials and Science Advances and other high impact journals as well. Here's some additional work that Morphogenesis has developed this cell spheroid printing that really was the foundation of Organovo, really the first large-scale 3D bioprinting company. This is work by Christopher Chen, where they had sacrificial sugar filaments, very similar in the idea to the work done by Jennifer Lewis. And very recently, Jordan Miller published last year in Science using not an extrusion-based technique, but a light-based technique of 3D printing that allowed creating a PEG or a GELMA block, but basically being able to pattern complex microfluidic networks inside, and had a very elegant example of an alveolar-like structure that could do gas exchange. So these are all amazing examples of 3D bioprinting with soft materials, but these are all doing something slightly different than what we wanted to do. We wanted to be able to directly 3D bioprint the soft materials. These are all kind of ways of getting around the challenge of directly printing these soft materials in different kinds of ways, right? So we really wanted to take that idea of the plastic printer that's just directly extruding plastic. Can we do that with a hydrogel? Clearly that you can't do that in air, and so we took a different approach. So instead of trying to make a biomaterial that's printable in air, which typically means modifying that material, making compromises, instead, can we make an environment that essentially makes any material printable? So in another way of saying this is, let's decouple the printability from the bioink and put the printability into the environment. And then almost any material now becomes printable. So the way we do this is this fresh 3D bioprinting. So fresh stands for free-form reversible embedding of suspended hydrogels. And really the most kind of prolific example we have of this is using a gelatin slurry-based support bath. And so if you look at this schematic here, we have the needle, we have the yellow gelatin support material here, which is actually a gelatin microparticle slurry. We can drop the needle inside, and we print a second hydrogel inside of that support hydrogel. We can print the entire structure, and we do all this at room temperature, and then we raise everything to body temperature to release the print, right? So that means the gelling fluid bioink is embedded in our sacrificial material during the printing. The bath that we print into actually acts as a yield stress fluid, so it transitions from solid to liquid-like. That allows the needle to move through easily, but whatever we print to stay embedded in place. This bioink is now uniformly supported during printing while it gels, and very often we have a cross-linker in the support bath that helps drive that gelation process. And then when we're done, we simply melt the support bath again at a physiologic temperature and releases whatever we printed. Really, the key rheology of our support bath is that it acts as a Bingham plastic. So if we look at this plot here of shear stress versus shear rate, you see the Bingham plastic here, and we basically need to hit a certain shear rate, and then the support bath flows like a liquid. So I'm going to play this video just to show a real example of the printing process in a time lapse. But what you're going to see here is you see the needle. You see it's moving through that support bath essentially with almost no resistance. That's because as the needle moves, it generates enough shear force that the support bath flows around the needle as a liquid, but then rapidly resolidifies. But whatever we're printing stays embedded in place. In this case, we're printing an alginate. The alginate solution's in the syringe. We have calcium ions in the support bath. So as soon as that alginate comes out, it's immediately ionically cross-linked into a gel. And then when we're done, we're raising the temperature of the build platform. You can see the liquefaction goes from the bottom up, and it releases our prints. Again, we can do this between room temperature and body temperature, which is really a thermal range in which cells and most biologic materials like proteins have, excellent tolerance. So, one of the exciting things with this approach is that we can fresh print a really wide range of bioinks, and most of these can be printed together if they have orthogonal or essentially non-interfering gelation mechanisms. So just as an example, we can print collagen by printing an acidified collagen solution into the support bath, where the support bath is pH buffered, and I'll talk more about this in a second. We can print alginate like I just showed, and alginate, we would have the calcium ions in the support bath that do the ionic cross-linking. We can do enzymatic cross-linking by printing fibrinogen into our support bath, where we have thrombin, and then thrombin will cross-link the fibrinogen into fibrin through an enzymatic reaction. Or we can use more traditional UV-based cross-linking, where we, in this case, have a methacrylated hyaluronic acid. We print that into our support bath, where we have ergocure, and we have a UV light that drives photo cross-linking. And this can all actually be done in the same support bath, in the same print. And just as proof of concept, you know, here the red is collagen, the blue is alginate, the pink is fibrin, and then the green is hyaluronic acid. So we've been really excited that that Fresh has grown as a biofabrication platform a fair amount over the past 5 years since we first published this in 2015. So this is an example of collaborative work with Eben Alsberg's lab, where he created these microspheres out of alginate that have embedded mesenchymal stem cells. These are alginate that are ionically cross-linked, but it's also methacrylated, so they can be UV cured later. And so these can be cryopreserved and thawed on demand, fresh printed into a structure, and then exposed to UV to gel them into a structure. So it's this really cool cryopreservable UV cross-linkable cell-based bioink. Tal Devir's lab at University of Tel Aviv last year had a nice publication using an alginate-based support bath, where they printed a heart-like structure. And then just this year, Brenda Ogle's lab at the University of Minnesota in Cirque Research had another heart-based fresh printed construct. In this case, we're actually to show a closed chamber that could generate pressure and PV loops. So really excited that these labs have been able to take this technology and also be successful with it. What I kind of wanted to talk about, though, is our latest results with fresh bioprinting. It's published in Science, again, about a year ago, a little bit more than that. I really focused on printing collagen, and we really wanted to focus on collagen because it's really the foundational ECM protein in the human body for every tissue and organ. And, you know, we figured if we could start to really print with collagen, can we start to really print things that don't just have really cool structure, but can they also now start to demonstrate true biomimetic function, so that they function in a way that's similar to the real tissue or organ? And so if we think about that goal and really trying to, again, move this towards organs one day, how do we get there? The key things we wanted to hit upon were, A, can we print at volumetric scale? So really, you know, single to tens of cubic centimeters, really large structures, you know, at least for state-of-the-art bioprinting. Number two, can we have mechanical function? We really looked at the heart valve to show this. Can we start to address this challenge of multi-scale vasculature from capillaries on up to, you know, arteries and veins? And then finally, can we do dense cellularized tissue? You know, one of the challenges with a lot of bioprinting is that cell-laden hydrogels typically have a relatively low cell density of tens of millions of cells per ml, you know, along with the biomaterial to get printability. But real tissues, muscle tissue, liver tissue, other tissues have extremely high cell densities, hundreds of millions of cells per ml. So how do we achieve that? So the first example I wanted to show here is of the heart valve. So this is a CAD model of a simple Trileaflet heart valve. And what we did was we 3D printed that at basically full adult scale. So about 3 centimeters in diameter, entirely out of collagen. And you can see that here in a couple of different views. You can even see the build line. So again, this looks very much maybe like an FDM plastic print, but this is 100% collagen. Now, what we did was we tried to mimic the way collagen valves are used clinically, which is where they take a porcine valve, they strip away the cells, they decellularize it, and then they fix it in glutaraldehyde. So we use the same process. We use basically the same fixation process on our valve, basically to make it stronger. And while these have not yet been in vivo, we put this on a flow system that simulates adult flow rate and pressure. And then here it is being imaged with an endoscope. And so we were able to create these functional collagen-based heart valves. And then just to kind of give you an idea of what the physical properties are, here's just a video of that valve being handled in air. And so this is, again, a really robust collagen-based valve that we have full control on the design. And so future work is going to be looking at improving the performance of this valve further, and then getting this in vivo in a large animal model to look at the ability to actually function as a valve in the actual heart. And so that's all on the slate for future work. The second thing I mentioned is actually creating dense cellularized tissue that has some level of function. And so the way we did this was we had basically a two bioink or biomaterial system. We had one syringe that's just collagen, and another syringe that's a cell-based ink that has cardiomyocytes. So these are human stem cell derived cardiomyocytes and fibroblasts at a very high density of about 300 cells per ml. And what we did was print a simple ventricle-like model, kind of like this open ellipsoid where the outside and inside shell are collagen, and then in between is a layer of these cardiomyocytes and fibroblasts. As I mentioned, the collagen is actually printed by having an acidic collagen that is rapidly neutralized. But we did separate these because we need to have a very high density of cells that have functional muscle tissue. And so one of the first things we did was actually print this cell ink and then this collagen ink next to each other to make sure that we could neutralize using our buffering system, the collagen, very rapidly, and that would not, the acidic, the acid in the collagen would not damage the cells, and indeed it does not. Here's some live dead where we can see high cell viability. Here's the simple model of this ellipsoid of this ventricle. It's really about the size of the end of the pinky. Here it is printed out of the collagen and cells, and then here's actually a staining of the cardiomyocytes in this wall of this ventricle stained for the sarcomeric alpha-actinin, and we can actually get a general direction of alignment in the print direction, which is really cool. So we can control cell alignment through some of our printing technology. So to look at function, what we did is we took these ventricles, we then looked at them under the stereo microscope under fluorescence using a flow-4 calcium-sensitive dye. And for muscle cells, when the cells contract, calcium rushes into the cell, and that's actually visible as an increase in brightness. So if I play this video, what you'll actually see is this wave of electrical activity that propagates spontaneously around the ventricle. That's spontaneous, and here it is actually under field stimulation. So we have some platinum electrode wires outside the field of view, and they stimulate the heart at a constant rate. Now we're gonna flip and look into the ventricle from the top. And again, this is spontaneous. You can see the wave of activity, again, going around the ventricle. We're then going to pace it. And then actually what's happening during pacing is due to the synchronization, we actually have wall thickening and decrease in interior chamber volume. This is actually the first time anyone had been able to achieve wall thickening in one of these engineered ventricles. And that's really exciting because that's the same way the heart actually functions. And so even though we're far, far below the level of function of the actual ventricle, we can at least start to function in the same way in terms of wall thickening. And so we're really excited that, again, this obviously can be improved much further than where we are in this video, and we're already doing that, building kind of these improved ventricle models in the future. The next thing I wanted to touch upon was basically vasculature and really this challenge of trying to build vasculature across length scales, right? As we know, we go from arteries and veins down to capillaries. We're talking about, vessels that are many centimeters in diameter down to vessels that are really, maybe even as small as 5 microns in diameter. So how do we do that? Right? We also have this complex three-dimensional architecture, right? Branching architecture. How do we rebuild that? That's really difficult as well. And then obviously the materials are soft. They're constantly remodeling. And obviously all this needs to be created in a system that's perfusible, right? So the pressure drops between levels of branching really needs to be correct for this whole system to become perfusible and to function properly. So those are some pretty high bars, I think, to hit. So how are we approaching this? How are we tackling this? So, I think there's a fundamental question about what do we need to print, and then what can biology do for us? So what scale can we fresh print? And then if microvasculature is required below the resolution of fresh, do we need to print it or do we not need to print it? In other words, can we rely on basically cells to build the microvasculature for us? And so that's really what we're doing. We're fresh printing from about 100 microns on up to larger scale, and then we're really relying on cell-based processes and geogenesis to basically build the microvasculature for us. And also trying to address the challenge of how do we keep cells alive long enough to have this microvasculature actually be built. So I'm going to want to show you kind of two examples of how we're approaching these different length scales. So the first is really the level of printing the vasculature directly, or at least vascular-like three-dimensional branching networks. What you see here is an MRI image of the human heart. You see the left ventricle in purple, and then you see the coronary arteries and veins in red and blue, respectively. Now, MRI is really exciting because we can get a patient-specific model of the anatomy, but clinical MRI has a resolution of voxel size on the order of about 1 millimeter, and a lot of the vasculature is below that. So how do we recreate a vasculature that we can't image? So what we did is we decided to use computational methods to generate vasculature and then try to print that and then perfuse it. So again, as I play this video, what you see here is this MRI image, this segmented image. You see the left ventricle. We're going to then focus on that left ventricle. What we're doing here is creating a distance map where red is near the artery and blue is further away. So we're looking at just the arteries now. We then created a vascular network using a Voronoi lattice structure where the vessels get smaller and branch more the further you get away from the arteries. We're then going to focus on a segment of this whole ventricle. This is where the root of the coronary artery is, as well as some of the surrounding vessels. Where basically it's about 2 by 3 centimeters. We're going to basically isolate just that. So the red here is the image by the MRI, and then now these smaller vessels is what we computationally generated. We're going to take that, create an STL, and now 3D print this entirely out of collagen type 1. We perfuse that, and we try to image it using fluorescence, but it's hard to see a lot of the internal structure. So what we did next was actually to clear this, clear the collagen so it becomes optically transparent. And so what you're seeing here is a real-time perfusion video of a blood substitute into this multi-scale network. So into the root here, larger vessels, smaller vessels now. It's actually leaking out the periphery because, again, this block was kind of just cut right out of the whole ventricle, so those vessels are open. But what you'll see us do now is kind of move this video forwards and backwards in time so you can kind of see, again, this interconnected multi-scale branching network be perfused. And if we zoom in here just on the very center, you'll see some of the smallest channels being perfused, and the smallest one right in the center actually goes down to a 100 micron diameter. So again, this shows that with fresh, we can really start to build these multi-scale vasculatures, at least vascular-like networks. These are not yet cellularized, but entirely within a collagen-based ECM system. So this is what fresh can actually print directly. So what about going smaller? So by going smaller, as I mentioned, we're really looking for cells, endothelial cells, pericytes, other cell types to help build that vasculature for us. And so the way we looked at this was to take a disc of collagen, about 1 centimeter in diameter, loaded with VEGF and also some PDGF, and either cast that into a disc shown on the left or create a disc the exact same size but fresh printed shown on the right. And these were both implanted subcutaneous in the mouse for 10 days. You can see just by gross examination that the fresh printed construct has much more vascularization. When we do cross sections and stain for H&E and also CD31 for the endothelium, we can see that we get cell infiltration in both, but only in the fresh printed construct at this time point do we have robust vascular ingrowth. The reason for this is actually by purposefully incorporating the gelatin microparticles that make up our support bath into the collagen as it's being printed, so that when we melt away the support bath and we melt away those gelatin microparticles, they leave this porosity behind, and you can see those pores that are left by the dissolving of the collagen. And just to kind of show how this is anastomosed, we did a lectin tail vein injection in the mouse. Basically, everything in all the vasculature of the mouse becomes lectin stained, and this is the construct that we removed and did some whole mount imaging on. And you can actually see this robust vascularization with some larger vessels up to about 100 microns in diameter, which again matches up with about as small as we can print, down to these capillaries that go as small as 5 microns in diameter. And so, again, while we have not yet figured out how to marry these two levels of vascularization up directly within a single print, that is ongoing research in the lab right now to do that. But kind of shows the potential of the technology at both different length scales. And then finally, I want to talk about going big. So we've always been inspired by decellularized organs as a amazing kind of proof of concept that if you could build, or if you had the ECM architecture with the right composition and structure in 3D space, and you put the right cells back, you start to really rebuild complex tissue. We just saw that decellularized organs, however, have a lot of challenges with recellularization, especially as you go bigger. So the vision was then to be able to take MRI images and use that to directly fabricate ECM-based scaffolds that were like decellularized organs. So we're not all the way there yet by any means, but this is one example where we took the adult MRI segmented heart image on the left, and we printed that not at full adult scale, but at neonatal scale, so about 5 centimeters top to bottom, entirely out of collagen. We were able to then take that heart and actually run it back through the micro CT. And I'll play that video here. So this is the heart that we printed. And again, in the micro CT, it looks like there's some holes in the wall of the heart. That's actually just more of a contrast issue, just because those areas are thin and didn't really resolve very well. But as we rotate this heart, you can see we can really recreate almost all of the detail that is in the MRI image. And now as we look down into the main arteries here, we're going to actually go slice by slice, and you'll be able to see now the different internal structures. So you see here the left and right atria, the pulmonary valve, the aortic valve, and then as we move deeper into the heart from top to bottom, you'll see first, obviously, this infill structure in the wall, but then also the left and right ventricle and the trabeculation inside. And so this, obviously really points to our ability to really start to create, complex 3D volumetric anatomic structure. And again, while these are not yet cellularized, because that actually requires a tremendous number of cells that are still difficult to produce, the idea of producing the scaffolds itself is really starting to be addressed. And we've I think, taken this all the way now to full organ scale, as I mentioned on the very first slide. We just published this last week or the week before in ACS Biomaterial Science and Engineering. But as I play this video, this is, again, a full-size heart where we basically took a small bucket, if you will, filled it with our support material, and printed the full-size organ. This one happens to be out of alginate. We showed that the mechanical properties can be tuned to be tissue-like, and that you can also begin to think about suturing this material and perfusing the vasculature. And so we think this is, really quite exciting in terms of thinking about how large you can go. And then, I wanted to talk about some of our work that'll be coming out in the near future, moving this more towards microfluidics. Again, since we can create these vascular-like networks. The idea that we can have a collagen-based microfluidic platform rather than a PDMS or a thermoplastic-based platform is interesting from the standpoint of creating more realistic models. So here's just a few examples of some different structures we've been building. Here's just some different single, dual, quad, and helical channels. So again, these are very straightforward to print in our system out of collagen. We have these in these perfusion systems. Here, these are not quite microfluidic. The diameter here is about 1.4 millimeters internally. But again, here's the single channel. Here's the dual channel being perfused. The wall here on these vessels is about 300 microns. Here's the quad channel, so you can kind of see some of the branching we have in this area. And as we play this, you'll actually see some of the bubbles that are perfused through as the perfusion starts. Then we also have here the helical channel. So we don't just have to produce these simple linear channels, we can be curved in 3D space really, however. And so the printing fidelity and the ability to print collagen-based systems that are essentially, watertight is quite good. And so we've also been improving the support bath. This is, again, some additional work that'll be coming out in the future. This is our transparent fresh support bath. Not a completely transparent, but pretty transparent, where we've been able, anyone who's used normal fresh knows that it can be a little bit translucent and a little bit hard to see through. But here is a straightforward collagen-based device that we're printing. It's actually a test piece to see what our resolution is that we can achieve. So it's this block, about a centimeter square, a little smaller, with 500 micron down to 150 micron with channels. And we've actually implemented an OCT, so an optical coherence tomography imaging system, so we can get rapidly 3D images of our scaffolds as we're fabricating them. And this just shows you the routine, resolution we can get here. This is 100 microns. On our newer printers, we can get down to about 100 micron with really, really excellent fidelity. So that really highlights the work we've done. I want to really thank the folks in the lab that have made this all possible. I have a really talented group. Josh Tashman was key on developing the cleared fresh support and a lot of our microfluidic printing. Andrew Hudson did the valve printing and has been doing those perfusion studies on some of the microfluidic work I showed. Andrew Lee really drove the work on the 3D bioprinted ventricle. Dan Shaworski is doing the work on the microfluidics, the OCT, and a number of other areas that he's contributing to. Iman Murdamati was a master's student who's now at the University of Maryland, who led the work on that full-scale bioprinted heart. And then obviously we have a lot of other talented folks doing work that I wasn't able to share today. We're on Twitter and we have a website, and I'd like to thank the funding support from NIH, NSF, Department of Defense, American Heart Association, Human Frontier Science Program, and then obviously the disclosures. I am co-founder and the CTO at Fluidform. I'm also on the advisory board on BioLife 4D, which is working on a bioprinted heart. I did want to, before I finish, cover two kind of things that I love to share. I think they're both exciting in different ways. The first is our 3D bioprinting open source workshop. So we actually have done all this work on desktop plastic printers that are open source. We created open source designs to basically turn them into bioprinters that we've published on. We've released to NIH 3D Print Exchange. And we also have a workshop we now run. We ran it in basically December of 2018 and 2019. We're not running it this year. We'll actually have a virtual workshop that we're going to be posting information about soon. That'll probably be in January or February. But basically, you can come to CMU, build your own open source bioprinter, and take it home with you. And we've really seen this as another way of trying to grow the number of labs that can be successful in bioprinting. This is actually just a subset of labs that have either worked with our lab directly or attended our workshop. It's really been exciting. So we have groups from really all over the U.S. as well as Korea to Australia to Israel to Europe. And so this has really been exciting. And so if you're interested, please go to this website. It hasn't yet been updated, but it will be soon with information about our virtual workshop. And then hopefully once COVID is no longer interrupting or really disrupting our lives, we'll go back to the in-person workshops here at CMU. And then as a final piece, if you're interested in fresh printing, Fluidform, again, this is the startup that's been commercializing this. We have a sterile lyophilized version of the Fresh 2.0 support that you can just buy, hydrate, and use. And if you're interested in doing that, we've partnered with Advanced Biomatrix, Alevi, and CellLink. They all resell this life support material. So please go to your favorite either bioprinter company or supplier, and you can buy this to try it out. So go back to, again, thanking the actual team that made all of this possible, and I'm happy to answer any of your questions. Thanks. I think I'll end the screen share now.
Well, thank you very much, Adam, for your very, very nice presentation. You didn't see our faces, but we were all surprised to see your new technologies and new results. So very nice work, as always. So before we start the Q&A, I would like to make a few announcements. First of all, our next speaker is going to be Mr. Scott Phillips from Starfish Medicals, one of the largest biotech companies in Canada. So please make sure that you mark your calendar and participate in his talk. I have participated in his a few of his talks, and then it's extremely inspiring. And then also I would like to thank again our sponsors, Montreal Transmed Tech, and then also Cellink for supporting this talk. So with that, I would like to start with the questions from the audience. So we have the first question. The first view question is from Diaz. I hope I pronounced, or Diaz, your name correctly. So the first question is about the sterilization of the heart valves that you have made. So is collagen, is it possible, or let me just rephrase it. How do you sterilize the heart valves that you create before, let's say, you want to implant them in the body?
Yeah, so the heart valves themselves have not yet been implanted, but we have implanted other constructs. So, you know, just to, so there wouldn't really be much of a difference because they're still collagen. But really, first of all, we produce the support material under sterile conditions. You know, sometimes we make it ourselves. Actually, sometimes we actually buy it from the startup that we have because it's already lyophilized and E-beam sterilized, so it's really great for cell-based. So that's basically, you start with sterile support, you use sterile collagen, we do all the printing in the biosafety cabinet. So we've done that. We have done implantations, both subcutaneous in the mouse and then also a surface wound we're doing for some volumetric muscle loss studies in a large animal model. And we've used the approach in both cases. I don't think we've had any infections yet. Obviously, that is not as good as a post-sterilization process per se. But, you know, it's actually difficult to sterilize collagen without altering its properties. And so this is just a challenge, I think, with any bioprinted ECM or bioprinted ECM in cells, right? There's not so many post-sterilization processes available. So it's really about maintaining sterility during the manufacturing or fabrication process.
So yeah, I mean, I agree with you. And then we're dealing with some of these sterilization issues with the hydrogel that we're working with. Yeah. And we tried gamma sterilization, and it completely changed the, the structure of the gels that we're using. But I mean, that could be a challenge, as you mentioned. Let's say that like towards commercials or selling these heart valves to patients, not to patient, not selling them to patient, but using them in patient, because eventually the ultimate goal is to make these heart valves and then giving it to clinicians so they can just place it in the implant it in the body, and then you probably need many of these heart valves. So what are the challenges besides these sterilization approach that you see, or what are the barriers against bringing this into clinic, the heart valve technologies?
Well, I mean, I think I think there's two big issues with the heart valve, right? Is, can we get the mechanical performance that we ultimately need, and can we get the biologic response that we want, right? Because what we're trying to, our vision, right, is that we get the biologic performance of the current biologic valves, which means clinically patients do not need blood thinners, right? That's the big advantage. Yeah. They can also be delivered through transcatheter approaches, endovascularly, right? So that's also great. You don't need open heart. So we think we can achieve that. I think we have some advantages there because we can tune the biomaterial we're printing, right? So we can print collagen, but we can think about adding additives or other compounds that might slow the calcification, which is the major risk, right? We don't necessarily have to use glutaraldehyde, which is known to enhance calcification. Yeah. So we have, I think, some options there to improve the material. Now, that's not our strong suit per se, like in our lab, but working with others who have those expertise. We also like the idea that because we designed this in CAD, we can manufacture it to potentially better fit for transcatheter delivery. It better folds up, right? Because otherwise people are trying to make other things work that are difficult to do, like an existing valve, or whether you're using pericardium that you sew, which is another approach to doing this. We also think, in a more advanced valve, you could potentially do it patient specific, right? Because when you use a porcine valve or even the standard valve, it's not a one size fits all, but there are set sizes, and not every patient anatomy is different. So can we think about in the future using computational modeling to determine what's the optimum valve design for the best flow condition in that patient, which could also even help with things like calcification, right? Can we actually then design a patient-specific valve and then manufacture that, getting the manufacturability you get out of the printing, but by making out of collagen, you get that collagen biologic performance, right? That's the vision. Now, achieving that, again, comes down to actually getting the right material properties, getting the right mechanical strength, actually figuring out how do you, through a regulatory process, get a valve that is unique to each patient. Because typically, right, when you manufacture a valve, you do a lot of them and you take a subset for destructive testing, but that might be really expensive with a patient-specific valve to do it that way. So what kind of in silico testing can we do? What is the validation quality control process from a regulatory standpoint as well? That all needs to be worked out, right? That's why, these things are going to take some time. Yeah. But I think we've all seen this field is accelerating in terms of what we can do. Yeah. And we're starting to get the investment as well, right? Because all of these technologies, I don't know if it's so much an if anymore, it's more of a when. And a lot of the when is a financial, there's a lot of, what are the resources that are being brought to bear to translate some of these technologies? I think that then is answered by what's the profit, what's the commercial, right, profit driver, the market size, right, for these devices. And so I think we're still searching for that torchbearer. Like, what's the first thing that kind of is the sweet spot of like, oh, bioprinting is a killer advantage, right? But there's also a large enough market, right? And we can make them cheaply enough. And then the regulatory hurdles are not too high. I don't think the field has quite gotten there, but I think we're rapidly converging. So. Okay,
that's great. That's good. That's promising and encouraging. That's awesome. But along the same direction, do you see these technology, especially the fresh bioprinting, which enables creating complex tissues? Do you see them being used by clinicians in the operation room? So they have this printer in the room. They have all the MRI images from the same patient that they're going to operate, and then while they go or prepare the patient, they'll just hit press print, and then they print the heart valve and then take it from the bath and then put it in the or implant it in the patient. Is that something that you envision happening anytime soon?
Not anytime soon. I do think that is a potential option. I mean, I do think one of the advantages with medical imaging is that we don't necessarily need to have it as that acute of a process, right? Which might open up more kind of manufacturing options and again, like sterilization options and other things we might be able to do. But, I've always thought that the idea of in situ, like in the patient printing, is pretty interesting. We're not working in that space, but I thought that's a really kind of an interesting idea as well. But, I think, outside of a very acute, like immediate, surgical need, we have time. One of the problems, as we all know, with 3D printing sometimes is time. So, right, if you're printing with cells, it's a huge issue, right? Because cells have a pretty limited viability window in suspension, which is how we print them. It's a few hours, 2 hours, 4 hours, maybe a little longer. There's some tricks we can do, varies by cell type. So how do you solve that? And we're looking at different ways of speeding up printing. That is an issue with size, but also obviously with extrusion, right? The finer the resolution, right? The more time it takes to fill up the same volume, so the longer the print. Yeah. And so I know a lot of people are always excited about, oh, what's the best resolution you can get? Well, I'm like, I don't know. In our system, we got down to 10 microns. That's great. I would never print a scaffold at 10 microns because I don't need to. It takes too long. Yeah. And that resolution's probably not required for a lot of applications. It may be for some though. Right. And so I think, there's all these parameters that matter. But, for example, we're printing scaffolds right now for volumetric muscle loss that have on the order of 10 to 20 milliliters of bioink. So pretty big. Right. So about 10 to 20 cubic centimeters of volume. And they take about 8 hours to print the way we want to right now.
Wow.
Right.
Okay.
So, they're for, so we're basically creating an injury, doing a CT scan of the animal, and then printing it and implanting that scaffold the next day to help guide regeneration.
Mm-hmm.
So, but yeah, those are some of the challenges we face.
I see. So how long did it take to print those heart valves?
The heart valves, I believe, were a couple hours. Mm-hmm. Not that long. Again, it's a little bit of a resolution game. The collagen, I always tell people, and this shocked us more than anyone else, the collagen is actually the easiest thing we have now to print. Like, it works so well. Using this pH neutralization in this approach, we get better quality, resolution, fidelity out of the collagen printing than we actually do out of anything else.
Yeah.
So, we're really excited by it, because hopefully that helps a lot of people. So.
Yeah. Okay. That's good. That's promising. In terms of time, because time has, as you mentioned, it's always, especially when it gets bigger, the construct gets larger, then it takes a few hours to print. But I'm sure that there will be. Many smart people are working on in the field of 3D bioprinting. I'm sure you will find basically a solution to that problem. So the other question is asked by Mehnush. Probably we already covered part of it, but the question is about the main challenges of using fresh method in 3D bioprinting of soft materials.
The main challenges, it was we developed it to solve challenges, but like any technique, it has its pluses and minuses, right? That's just technology, right? So, it's temperature sensitive, right? So you print at room temperature, you raise it to body temperature to melt the support. Now we did that intentionally because it's an extremely gentle release, right? And so we've found that there's a lot of different support baths that have now been developed out there. They all have their own advantages, but we haven't seen anything that's as gentle of a release and maximizes kind of viability and structural integrity like the gelatin does. But if you need that structure, that support material to be around longer, obviously you go in the incubator, you melt it. So it's not there for that purpose. We've also had some issues producing the support bath over time, because it turns out that the gelatin that you buy is so incredibly variable, lot to lot. Like, it's the worst thing out there from almost any bioreagent we've used. And so we've really had to get really deep in the weeds on a specification for the gelatin. Far below what you would ever see on a normal manufacturer's label. To be able to effectively get good support material. That's actually that's the kind of thing that the company solves, right? The company solves that, so no one, if you want to buy it, you never have to worry about that again. But that has been a challenge that we've largely addressed at that point, but that's always been difficult. And I think that probably holds for other support baths that are out there. But we're mostly leveraging the advantages of it right now. There's not too many disadvantages beyond kind of the obvious ones you see. Oh, and I would say another one is that the support bath is made out of microparticles. So those microparticles can actually, if you look in our science paper, I didn't show any images of it in the talk, but you can see little dimples on our filaments where those microparticles were. So that might be an issue for some printing. Like I said, we actually turned that into an advantage, because for our microvascularization work, we actually incorporate those microparticles in purpose into the print. And so when we dissolve the gelatin support bath, we have these little micropores, and it actually turns out to be great for cell infiltration. But I think that's like most things, right? It can be a feature or a bug depending on how you look at it. So
yeah, cool. So I just have one more question about the challenges. So have you faced any issues with the premature cross-linking of the ink at the tip of the needle using this method, or you didn't see that as an issue?
Yeah, the only time we see that is when we are doing multi-material and we switch nozzles and we stop extruding out of a nozzle, right? And so we show pictures of it in some of our papers, but we've basically developed a system when we're done printing, we put it into a DI water bath. The needle that's not printing is hanging out in the DI water bath, and that basically dilutes enough of the crosslinker that when we go back to start printing again, it hasn't clogged up. But there's one thing that we do with our printing typically, and we don't use a pneumatic extruder. We use a direct lead screw driven, plunger driven, syringe based extruder, and so it's constantly retracting. And so you have a lot of mechanical movement back and forth. I think that probably helps prevent a gel clog as well. So,
yeah, I agree. Because I was a little bit worried about alginate, the alginate ones that you print, because alginate will be in contact with calcium chloride, and then there might be some diffusion from the calcium. But
I wouldn't say we've never clogged a nozzle. I mean, we definitely on occasion have clogged the nozzle, but it's actually quite rare. And if we're printing it at a high enough speed as well, that tends to solve that. So
yeah, that's brilliant. So the other question is from Diaz again. I mean, I don't know if I pronounce his or her name correctly. So how did you synchronize the spontaneous contractions of myocytes in the heart valves?
So the myocytes were the ventricle, but I'll talk about the ventricle. That was the cellularized one, but that's just field stimulation. So outside of the view of that video, there are 2 platinum wires in the solution, and we basically send a square wave of voltage across them. It's kind of like when you've seen someone ever defibrillate the heart, where you send a large shock to kind of synchronize the heart to contract at once. That's essentially what we're doing. So it's a pretty common approach to get synchronized contraction in engineered cardiac tissue.
Yeah. The other question is from Mark Prier. Can you comment on the maturation state of your bioprinted ventricles? Meaning, does it show positive force frequency?
Well, so for that tissue, we didn't measure force, so I have no answer. My guess is that they're relatively immature. We've got another paper that's actually up on bioRxiv right now. That's not bioprinting, but it's actually, we developed a little engineered tissue construct where we actually look at loading and using tunable mechanical loading to try to drive some functional maturation, where we can look at force frequency relationships. So I know that loading is important, and this construct we created, because this is open ellipsoid, we're not controlling loading. The next generation of this system that you'll see from us is a closed chamber system, right, with valves, so that we can actually pressurize the system and actually put in a pressure catheter transducer and do some of that work. So I don't know the answer. My guess is that these are immature. It was really hard just to make that ventricle, to figure out how to do it. You may notice that we printed the collagen and the cardiomyocytes separately. Yeah. We went through a lot of different design iterations before we hit upon that. We realized, for a long time we were printing cell-laden hydrogels, because honestly, everyone in the field prints cell-laden hydrogels. That's just what you do. We realized the density of cells was so low and the quality of tissue we're getting out was so poor. We need way, way, way more cells.
Yeah.
How do you do that? You either print higher density, or you need your cell-laden hydrogel to compact to densify. It's got to be one or the other. We went with the high density bioink. By doing that and kind of using the collagen as a separate ink to create more of just a supportive shell, that's how we got to a tissue that at least could contract the right way. But again, as many people know, ES or IPS derived human cardiomyocytes tend to be quite immature. My guess is that these are quite immature. The conduction velocities we were getting were only about 1 centimeter a second, 2 centimeters a second. That's quite slow. So again, a lot of room for improvement. We're actually really excited to try again the devices we're building next to do some of this mechanical loading to try to drive some of that structural and functional maturation.
I see. Great. And then speaking of high density cell printing, have you? I mean, you have the results, but before that, have you faced any issues with the shear stresses being applied to these cells during the printing process? And how did you fix the issue? if you had any problem with that.
Yeah, I mean, our problems were primarily around, well, there's a couple different problems. One is just, if the nozzle is too small, obviously, the smaller the nozzle, the higher shear stress. I believe we were using nothing smaller than 150 micron diameter nozzle, right? So we have essentially a resolution limit with the cell printing. Also, you need to make sure that when you have a high density cell bioink like we're using, it's really a cell slurry. It's basically if you ever passaged cells and made a cell pellet. Yeah. We basically have a cell pellet with just a very little bit of extra liquid.
Yeah.
The cells cannot stick. If they stick, then they become bigger agglomerates and the shear goes up. So we actually have a paper we're working on where we're using both temperature and some other tricks to slow down the cell-cell interactions to both improve that and also prolong the working window of the cell ink. So, instead of 2 hours, can we stretch it out to 4 hours? It's still not game changing. If we go from like 2 hours to a day, that would be game changing, but we can at least we can kind of double the working time, but it also decreases the shear stress. And so, obviously it's obvious things like interfering with cadherins, interfering with integrins, the way that the cells stick to each other. If you can slow that down, not in the way that damages the cell, but just enough transiently, you can get some advantages. So there's multiple issues, but yeah, shear stress is a killer. There's other groups that have developed bioinks that are meant to actually just better shield the cells from the shear stress as well. And so, that's interesting as well. I think there's still a lot of improvements to be made there though, overall. So,
well, I would love to read about your other tricks in the paper that are coming out, because this is very interesting and exciting, and I'm looking forward to that paper. And have you ever thought about maybe encapsulating the cells? You showed some of the work with Evan, and then but are you planning to use those hydrogel-based microparticles? So you encapsulate the cells in those hydrogel micro capsules and then print them as a way to avoid the effect of shear stress on the cells?
I think we're still trying to figure out and I think that this is like the field is trying to figure out what is the best thing to print.
Yeah.
I don't think there's an answer.
Yeah.
It's probably also application dependent.
Yeah.
Like, what are you trying to make? Also when we think about speed, those bigger cell spheroids or those encapsulated cells are also really interesting from a speed standpoint.
Yeah.
Because they're bigger, right? We have a bigger nozzle. Whatever we're printing, we print faster because we've got the bigger nozzle. So that's another way to try to achieve. So we are trying to balance these different concerns. And I think, so we're not specifically using that, but we do have a project where we're, we are trying to figure out, is that cell slurry, is that better than cell spheroids? Like, what's the right thing to be printing? And I think the answer is we're really going to ultimately just want to have a toolbox where for whatever we're actually trying to make, there's probably an ideal answer for that approach. Depending on size and speed and complexity of the tissue and so forth.
Well, great. These are great projects for the future of fresh 3D printing. So I'll let Human ask a few questions if he has, because I know he is in this field. He knows more than me about cardiovascular tissue engineering, and he uses bioprinting. So I see that he's going up and down to ask questions. So I'll let him ask a few questions before I continue.
Thank you so much, Mohsen, and thank you, Adam, for the nice presentation. I have been following your work since you started the fresh 3D bioprinting by publishing ACS Biomaterial Science and Technology, the first PDMS printing, and after that Science Advances paper and then Science paper. So it's really exciting to see this technology is growing fast. So as you mentioned, so there is no way, I just want to clarify this, there's no way to print cells with collagen, since collagen is acidic, right? So for the heart valve, we should always have the scaffold printed and then later on seeded with the cells, right? Because we do also fixation of at the end of the printing, right?
Well, so the collagen printing that we've published is the acidified collagen. There are ways to print cells and collagen together if you would want, but you have to obviously use a different gelation mechanism than pH neutralization, right? We found that we got excited about that because the quality of the collagen that comes out of that process is pretty phenomenal. It actually allows us to create things like durable heart valves, which is typical. It's tough with hydrogels, right? And actually, to give you an idea of like how far we can take that, you know, that valve I think was printed with like around 24 mg per mil collagen. We're pushing that close to 100 mg per mil collagen now, right? And so the mechanical properties are even much better even before you do additional cross-linking. But those materials are actually brick wall, like that's a brick wall to a cell. Like collagen at that density, properly assembled at that concentration, like cells are not getting through that anytime soon. So, so it really just depends on what you're trying to do. So that might be interesting for printing more of like a cartilage, where you would want a low density of cells maybe embedded in collagen. And there's some ways we're thinking about trying to do that. But otherwise, the collagen is different. We are mixing in the collagen with some other bioinks, like that cell-based bioink that we're printing with hundreds of millions of cells per ml. Actually, that has fibrin in it. So that's how we gel that. There's fibrinogen in there. There's thrombin in the bath. And it holds the structure just long enough so that once you put it in the incubators, the cells can spread out and connect and stabilize themselves. But I think we all know, like, fibrin is a really weak gel, especially compared to collagen at similar mass, right? You know, we use collagen at maybe 10 mg per mil. We typically use fibrin much higher. But yeah, there are ways to, again, get the collagen as the matrix, but you can't use this acidified approach to do the gelation. You could use methac-related. You could also do some thermal-based approaches if you can get the timing right. So.
And the other thing is the fixation. I mean, if you have the cells and you fix the cells to increase fix the construct to increase the mechanical property with glutarate. So that also is kind of bottleneck or limitation.
Yeah, exactly. So our fixed valves are not meant to actually be like regenerative. They're actually more of like a medical device, really just like normal porcine valve. It's just, can we make them better by, again, matching patient anatomy, optimizing the fluid mechanics right through the valve, making it maybe easier to fold for transcatheter delivery, those kinds of things. For a regenerative valve, you can't cross-link it. And in fact, it would be a very different kind of device. But even with that, I think you asked the question of like, how would you even want to cellularize a regenerative valve, right? You might think that do you need to endothelialize the surface? Because maybe that can happen once you implant it. But for valve interstitial cells, do you need to print those into the construct? I think those are questions we have with a lot of the tissues we're trying to create, right? How do we create them? What do we want to print as we go? What can we maybe seed later? Or if it's going to be implanted in vivo, what can it actually recruit from the host from a cellularization standpoint? Because, the reason our bigger hearts are not cellularized is that, we don't maybe have enough time to print it that big, but it's really just hard to get enough cells, right? The cost of doing iPS-derived human stem cell cardiomyocytes is very high. So for me to generate billions of those cells for a construct is basically just not feasible at today's cost and scale and ease of production. So that'll, I think, be solved, right? There's a lot of folks working in stem cell-based, expansion technologies, but today those are some of the limitations.
Great, thank you so much. And maybe one more question about like heart ventricle. So how far we are from having a heart ventricle pumping the fluid out of? You didn't measure the output, but I know that some people do it. But how far we are from that to have the bioprinted heart ventricle pushing out or pumping out the fluid?
We're not too far from that.
Yeah.
I wouldn't be overly impressed by the flow rates.
Yeah.
But we're not too far from that. At least like basic proof of concept being shown. Yeah, we're not too far from that. We're really, I mean, that's a nice proof of concept. We're really interested in once you have a closed pressurized system where you can control preload and afterload, right? Can we start to use that to help drive kind of the structural and mechanical maturation of the IPS or derived cardiac tissue? So that's really kind of what we're focusing on. It's like now that we have these tools, we can actually do the more of the science of like trying to understand what we need to do to build better muscle. There's obviously there's the matrix, there's the tissue stiffness, there's the stress and strain on the tissue, there's the different cell types, there's things you can add to the media. There's a lot of stuff
to be done.
So we're really focused on building a test bed so we can start to understand some of these things. So I think it's important, I think, to, at least from our standpoint, to build a test platform, pause a little bit, do some basic science and hypothesis testing to better understand what's going on, and then hopefully then take those lessons and then move that to the next larger scale of constructs. So
sure. Yeah, very exciting. So just before I pass to Mohsen, I see lots of nice comments in the chat box about your talk, and participants are really excited about your talk. And also, we got many questions in the Q&A box that Mohsen probably can moderate. Thank you so much.
Sure. Yeah.
Well, for sure. Again, it's very exciting and interesting to see that. A multidisciplinary approach in which engineers, cellular biologists, material scientists, they've come together and then develop this amazing technology that hopefully at some point it will solve the issue that that we have right now, a big clinical issue of basically organ transplantation. Heart transplantation is a major challenge, and then a major clinical challenge. And then at one point, I'm very optimistic that with all the efforts that researchers such as Dr. Feinberg and others are putting into this technology, at some point we will have these beating hearts from like patient cells, derived from patient cells, built in a lab, and that they can be implanted in patients and save their lives. So it's amazing how the technology is moving forward. And I would like to ask another question from the audience, which is from Mariam, and then she is asking about the types of hydrogels that you're using for in fresh bioprinting. What are the when you want to choose a hydrogel or design a hydrogel, what kind of characteristics are you looking for this particular printing approach?
Yeah, it's a really good question. So, the things that work best, that are hydrogels are things that when you extrude it, it's going to gel rapidly through some mechanism that you control. Otherwise, it'll just diffuse away, right? So that's really the major thing. And I think what's been nice is we've shown that it's, it could be, pH driven, ionic, enzymatic, UV. Actually, as a little teaser, we have a collaborative paper with the lab of Sarah Heilshorn at Stanford University that's accepted and hopefully will be out, in a matter of weeks, using a click chemistry-based crosslinker that's in the support path for a range of bioinks as well. And so that really shows, I think, that you need the crosslinker in the support path, ideally. And then, as long as it gels rapidly, you're good to go. Now, you can print other materials that don't gel rapidly if they're not miscible in the support path, right? So we can print PDMS, for example, even though it's not cured, because it's so hydrophobic, it's not going to go anywhere. It just stays where you put it. So there are other biomaterials you potentially print as well. And we actually have some work we're doing on a soft robotics project where we're printing cells and PDMS in the same system to build like a little soft robotic type device that's muscle powered. So that is feasible. So
cool, that's interesting. But speaking of diffusing, the diffusion challenge with the bioink, you mentioned that you incorporated growth factors in your bioink. Have you had any issues with the diffusion of VEGF, for example, from the bioink into the bath?
Yeah, it's a good question. So we typically use something to help with that. So in the case of VEGF and PDGF, it's fibronectin. So fibronectin is kind of acting as our glue, if you will, to help. Fibronectin binds to collagen. The growth factors bind to fibronectin. It does help retention. We haven't rigorously quantified it. I guarantee you, we're losing some, obviously, but that's one way we do it. You could obviously also tether it using other kinds of chemistries more directly into the system. So, but that's the approach we've taken so far. We've also done a lot with a decellularized matrix because most decellularized matrix is mostly collagen, so it prints with the same basic gelation mechanism as a collagen. And in that case, we do retain a lot of what we're printing, but obviously some of it you do lose through diffusion.
Cool. Go ahead, Juan.
Yeah, just to add to Dr. Feinberg's comment, just follow up. We use the fresh printing technology to print the bioelastomers, polyester-based bioelastomer. So since the bioelastomer was not rapid cross-linkable with UV, so we just try to integrate it in the hydrogel, keep it, and later on shine UV, and then it keeps the structure intact after we wash out the acrylic-based other kind of hydrogel than gelatin. So it's pretty very, very versatile technique to do different biomaterials, as you mentioned. So just to add that we did that and it worked. Yeah.
Awesome.
That's great. So we are running out of time. It's almost 10:30 Pacific time, and I'm sure Dr. Feinberg also, he's busy. I mean, he's running two teams. One is his lab, which is, you saw many students are working with him, and then also he is the CTO of a company, so it keeps him that position also keeps him pretty busy. I have the same situation, and I don't know how you guys handle these many things because I can't. I mean, so with that, I would like to thank our speaker, Dr. Feinberg, for his amazing talk. It's always great to hear the new technologies and the new advancement that you're making in the field of 3D bioprinting, in particular cardiac tissue engineering. Thank you. Also, I would like to remind everyone that our next speaker is Mr. Scott Phillips, the CEO and founder of Starfish Medicals, one of the largest biotech companies in Canada. Also, I would like to thank our sponsors, Montreal Transmed Tech and CellLink, for always supporting us, especially CellLink, who supported this presentation. So with that, also, I would like to thank the participants and then wish you all a great day or night ahead of you. Take care.
Thank you so much, Dr. Fine.
Yeah, thank you both. I really appreciate it. Thank you, everyone.
Bye-bye.
All right, take care.
Take care.